Welcome to the Ed Outreach Training Program online. Part D explores the plankton activity in the program When Invasives Attack. Plankton are defined as the small and often microscopic organisms drifting or floating in a body of water. They consist chiefly of diatoms, protozoans, bacteria, small crustaceans, and the eggs and larval stages of larger animals. Some aquatic species are adapted to feed on plankton, especially by filtering the water. Zebra and quagga mussels have a direct impact on the aquatic ecosystem, starting with filter feeding plankton out of the water. When mussels' waste is deposited into the lake, the net movement of energy is out of the water column, where photosynthesis is happening, to the bottom of the lake. This creates an imbalance in the ecosystem. The plankton samples will be obtained ahead of time by dragging a special net along the water surface. This is called the photozone, where sunlight penetrates. To prepare a microscope slide, use a pipette to place a sample of water in the center of the slide. 1 or 2 drops will suffice. Carefully hold a cover slip over the water sample and drop it. The water drop will spread out into a thin layer where plankton should be readily identifiable under the microscope.
The point of this station is to compare the diversity or number of different kinds of plankton in the 2 samples. Let's look at how to operate the microscope. Use the switches on both sides of the base to turn on the microscope. You can also use these switches to control the brightness of the display. Press and hold the button on the display until it beeps and lights up. This may take a moment. Insert the slide onto the stage between the slide clamps. Use the knobs on the left side to adjust the position of the slide. Have students begin by using the shortest magnification lens, which has a red ring around it. The focus adjustments are located on both sides of the stage. Use the large knob to make significant adjustments and the smaller knob for finer ones. When an organism comes into view, use the lens with the yellow ring, which has the next higher level of magnification, to view it in more detail. Make sure students take some time to look at both plankton samples. Try to spend the same amount of time working on each one. Make sure students record their data, including the name or description of the different types of plankton as they find them under the scope. They can use the identification guides at the station to help them, but figuring out the exact type of plankton is not as important as coming up with the number of different types in each lake sample.
Plankton identification is done by observing shape, color, and motion. By using a dichotomous key, students can identify phytoplankton, or plant-like plankton, or zooplankton, which are animal-like plankton. Recording data. This is the most often overlooked aspect of the activity when students start discovering unusual species. Remind students often, scientists record their observations so that they can be shared and communicated and ultimately used by others. Interacting with students. Help students to use the microscope properly. Jumpstart the ID process by asking guiding questions about organisms on the screen. Resist giving the answer. Encourage the process. Please direct any questions about your work as a volunteer with Ed Outreach Day to Barbara Leese at barbara.leese@aiu3.net.